Officially welcome to our second webinar from our webinar series together with NanoTemper, optimization of membrane proteins for cryo with the Prometheus Panta is this presentation called. My name is Philipp Harnig. I'm head of laboratory at Biotech, and my co-host today is going to be Dr. Ping Zhang from NanoTemper Technologies. Ping, it's always a pleasure to do this with you.
Me too, Philipp. And hi, everyone from my side. And it is really great to meet you all here today. So my name is Ping, and I am an application scientist working at NanoTemper. So I'm very happy to be here today. Philipp and I have got a great lineup, and I'm very excited to dive deep into some data with you.
Yeah, absolutely. Yes, this webinar is part 2 out of 4 from our joint series. If you have missed the last webinar, no worries, we get you covered. The introduction webinar, so part 1, is accessible either via the Biotech YouTube channel or the NanoTemper website. We're also going to send you all relevant information, so a link to it that you can directly access it via email in the aftermath. As soon as you're registered and attending here, you're also going to receive this email. So do not worry if you didn't watch it. You will also still be able to follow this. So for the next 2 future webinars, you can register very soon. Actually, right after this webinar is going to be possible. It's also going to be in the email. We're going to send you a link to register. Today's focus is on real-life data and what it teaches us about solubilizing, stabilizing, and in the end, ultimately preparing membrane proteins for high-resolution cryo. We're going to walk you through some case studies and introduce you to a workflow that we've developed in collaboration with Greg Dodge. He's formerly at MIT's Barbara Imperiali lab and now works at Amgen. And Barbara, Greg, and the team provided us with a data set that we're gonna analyze together today. Ultimately, those examples actually have shaped how we think about copolymer selection and validation. We're now going to start with introduction to the NativeMP technology, and then we're also going to talk about the NanoTemper's multiplexing optical methods before we then dive deep into the data.
Exactly. So before going into the case studies, maybe let's briefly introduce a platform that makes this work possible today. So first, NativeMP. So I know that Biotech has developed this platform as a universal solution for detergent-free membrane protein purification and stabilization. So I learned that each of these polymers from this platform has been designed and selected for specific needs of extraction and stabilization. So Philipp, could you please give us more details on the range of the copolymers this platform offers?
Oh, yeah, of course. I would love to. This is what I do, what I like to do best. So what makes NativeMP unique is that it is basically one technology for the entire membrane protein workflow, basically from gene to structure. It's a completely 100% detergent-free stabilization, purification, and ultimately the solution for all membrane proteins out there. At least this is what I experience here in my lab, doing all of these interesting targets. So the special ingredient in this diversity is the diversity of the copolymer families. So we actually have 32 different copolymers based on 5 different backbones. And this is what you can see. Wait, I'm going to put the marker on what you can see here. So those 5 different copolymer backbones are the SMA family. This is the classic styrene malic acid. It includes standard but also raft-based derivatives like SMA200, we're going to hear today about it, or also the SMA BZ series. Then we have the DIPMA copolymer class. It's also, together with SMA, the first copolymer class which was introduced into the market. Those are aliphatic analogs of SMA, and those avoid aromatic interference, so they don't have any absorption at 280. And they are also way more pH tolerant than SMAs. We have the ASTIs. This stands for polyacrylic acid co-styrene, and those have a greater charge control, and we experience that they're ideal for eukaryotic systems. We have the ultrasolute amphipole family. These are modified amphipoles that actually do form nanodiscs on their own, unlike the original A835. And then what we're really proud of is the Cubipole family. It is our own proprietary backbone, and this incorporates all known sidechain modifications which are out there in the market into one polymeric platform. So this is really nice. And in the end, what you have to do is you just take your cells or your lysate or your membrane prep, you throw the polymers on there in solution. They're going to self-integrate into the membrane, cut out those native nanodiscs, as we call them. And then after you went through the purification process, from then on, you can use them just as soluble proteins, basically. You're going to end up with very nice elutions. And this is an example here. You can use them as you are used to with detergent at 4 degrees or also at room temperature. We like to do this. And you're going to end up with a portfolio of different, of your membrane proteins stabilized in different copolymers. So this is what we call the screening. And the nice thing is that all of these copolymers that we sell, they are not just tested with synthetic lipids, but they are actually tested with real biological membranes. So this is why we say NativeMP is really a true real-world platform, because we do screen all of our copolymers with every target we work on, and so we know that those actually work very good. But yeah, before we dive deeper into the case studies, Ping, can you give us like a nice introduction into the Prometheus Panta and tell us why this fits so well together with our copolymer platform?
Yes, with pleasure. So Prometheus Panta is the analytical toolbox of today's workflow. This instrument was actually designed specifically to support comprehensive protein characterization, including complex formulation and stability decisions. So it is also very well suited for membrane protein research, especially for nanodiscs. So what sets Prometheus Panta apart is its multiplexing optical methods. So with just 10 microliter of sample in capillary, you get 3 independent but complementary readouts. So first, you see the dynamic light scattering. DLS gives you an immediate insight into particle size and homogeneity. So in other words, you will learn the hydrodynamic radius, polydispersed index, and aggregation behavior of your sample at an isothermal condition. And for cryo-EM, this is crucial because you know that as particles in the 5 to 20 nanometer range with low polydispersed index are optimal for grade preparation. And the highlight is not only we can measure DLS at an isothermal condition, but also in a thermal ramp. And this allows us to get, for example, the onset temperature of the sizing increase, which gives us one more important parameter to identify conditions that gives good colloidal stability of the nanodiscs. And then second, you see the nano-DSF method. So nano-DSF actually detects the intrinsic fluorescence from tryptophan and tyrosine residues to measure protein's conformational change. So it lets us to track protein unfolding, for instance, along a thermal ramp. And it is label-free method, meaning no labeling or modification is needed to your sample. And the parameters such as melting temperature Tm allows us to compare and select conditions in which the protein has good conformational stability. So last, we have the back reflection optics, which add another layer. So back reflection measures stability by tracking attenuation of back reflected light during the thermal ramp. So turbidity increase indicates the formation of large amorphous aggregates, and this method gives us another important parameter to today's workflow, which is the onset temperature of turbidity. So for membrane protein nanodiscs, we will be able to use this parameter to track irreversible aggregation of the embedded protein after unfolding. And what is especially powerful is the integration of all of these methods in a single simultaneous run. You do not need to split your limited sample across different assays or instruments for multiple parameters that you will eventually need in screening. And it is precise, label-free, sample saving, and fast, and it gives you the data you can confidently act on. So before we go into each of the case studies, I also want to introduce you to the general workflow using the NativeMP platform and Prometheus Panta. So starting from expression and purification using the NativeMP platform, you have 30 microliter of sample with each copolymer. And then 20 microliter was used for SDS page and Western blot. And then 10 microliter is loaded into Prometheus Panta to perform first step 1 size analysis, which is an isothermal DLS measurement. And then step 2 thermal unfolding analysis, in which all the aforementioned optic methods were applied to monitor the sample simultaneously along a thermal ramp. So here you can see on the right side, the multi-parameters that Panta can generate from this 10 microliter of sample in step 1 and 2. And then most interesting is step 3. This is up to you, the scientist, to perform an integrative analysis of all these parameters. And looking at these different parameters, you will be able to see a unique stability profile for each sample so that you can find the optimal candidate to scale up the production for cryo-EM or any other assays. So maybe to wrap it up, this workflow combining the grid selection of the copolymers from the NativeMP platform with the multi-parameter analysis of Prometheus Panta helps mixed membrane protein research a lot more efficient. So it will let you find the right conditions early on using just a minimal amount of sample.
Yes, minimum amount of samples sounds really good. That was a really great introduction thing. Thank you very much. So much biophysics. So I guess this is going to be what we see in all the 3 case studies we have. Is that right?
Yes. Yes, here's the introduction slide. Again, we want to thank Barbara, Greg, and their team for the collaboration on those case studies. And in the next slides, we will see actually how 3 proteins perform better with one copolymer over another. And you will find out how early screening with Prometheus Panta can pinpoint which copolymer protein combinations are stable, mono-dispersed, and cryo-EM ready.
Yes, exactly. So we have 3 case studies. We have WbaP, we have PglB, and we have PglC. And we're going to walk you now together one by one through the case studies. They're all somehow similar, but also very, very different. So they are all bacterial proteins. They are all glycosylated, but they are very, very different in their shape. So for our first membrane protein of choice, this is the WbaP. It's a protein from Salmonella enterica. It's actually a monotopic phosphoglycosyl transferase, such a hard word. It plays a key role in the O antigen biosynthesis. So structurally, this is dominated by transmembrane domains. It includes a re-entrant helix, and it has only a very, very small cytoplasmic domain. And we try to put this up here. So all this is embedded in the membrane, and just up here, these little parts and the parts down here are actually outside of the membrane. So you can say it's a tough one.
Can you elaborate a bit more why it is tough?
Oh yeah, of course I can. Of course I can. So WbaP, as I said, is a part of this large and diverse family of those monotopic phosphoglycosyl transferases, and those are basically enzymes which are essential for the bacterial glycan biosynthesis. And this includes the O antigen assembly in those gram-negative bacteria. So despite their functional importance, this protein class remains completely underrepresented in structural databases. Prior to Greg's work, which we can see here, and he actually published the structure of this in eLab. We're going to show you this later. There is no high-resolution, full-length structure which has ever been solved before him. So this is largely due to their very challenging nature, and this is really a great accomplishment.
Mm-hmm. And maybe for me and our audiences, what were the main challenges when it came to actually get a cryo-EM structure for this protein?
That's a very good question, Ping. So primarily, the protein is embedded into the membrane, and as I introduced it has very minimal soluble domain exposure, which complicates everything from expression over purification, and also, of course, the biophysical analysis. You have this ball which is embedded in the membrane, and you somehow have to get it out of there. So additionally, these proteins often interact very closely with very specific lipids or membrane microdomains, meaning they're very easily destabilized when you extract them with detergents, what's the classical way nearly everybody's still doing out there. And that's why WbaP is such a strong test case for any detergent-free purification platform whatsoever. And it's a really cool example which shows how powerful this combination of your NanoTemper Panta instrument and our copolymer suite is, and why this collaborative approach is really cool. Yeah. So Greg used our platform to screen a bunch of different copolymers, and here on the side you can see the SDS page results, which showed that actually multiple candidates solubilized WbaP successfully, and it was also very well possible to purify the protein in those different copolymers, which is already great. I mean, it's the first step to purify the protein, right, if you want to solve the structure. And these included SMAs and also several ST variants. But we have to be careful because SDS page alone or COMASI alone doesn't tell us the whole story, right?
Exactly. So that's why the Prometheus Panta comes into play. So here you can see on the left side the DLS analysis. It reveals that while many samples had bands on SDS page, only some produced more dispersed particles with hydrodynamic radius below 10 nanometer. So you can see here we kind of give them some names. We group them into the good, the bad, and the ugly. So I want to highlight here is like the DLS really helped to discover the ugly samples, because you can see these 3 samples showed no differences to the good samples on SDS page. But the DLS data tells you that they're ugly due to the presence of unwanted larger sized particles. And now maybe let's focus on the good samples shown by both DLS and SDS page results. And we can see, for instance, SMA 200, ASTY 650, and also the small BZs are among those.
Yes, exactly. And this is also where we come to the paper I was earlier talking about, because with SMA 200, Greg was actually able, from the screening pool, so from very, very little sample, to actually obtain the full cryo-EM structure. That's amazing. Like we have rarely seen something like this, that from such a small sample, you can see it's also possible to have the major success, especially now for those monotropic phosphoglycosyl transferases, out of the screening purification. So no upscale, nothing, just like this. He was very lucky from this, but you can see that this is possible.
Yeah, that's indeed amazing. And the Panta data also support that choice. So with the SMA 200, the nanodiscs were not only highly uniform in size, but also the thermal unfolding data showed really good thermal stability, indicating also good structural integration of this nanodisc.
Yes, exactly. So the combination of the clean SDS page, elution, the compact nanodisc size, and DLS, we say 10 nanometer for this kind of protein size is something we would hope to expect. And the thermal resilience made SMA 200 in this case a really, really solid fit. And so this is a perfect example why we stress these multi-parameter validation. The structure Greg obtained confirmed the protein was intact, it was properly folded, and this, yeah, it perfectly validates the workflow from end to end, basically.
Yeah, it is definitely a great case that illustrates how combining both the stability data with structural success creates a very compelling validation loop. And without the early DLS and now DSF profiling, I think it is very hard to identify SMA 200.
Yes, exactly. So yeah, especially this reminds me day after day when I use the device. We use it actually in the lab every day here. We're very happy to have it. Just don't pick based on the yield or just the SDS band intensity alone. It's really this downstream compatibility that truly matters if you want to do structural biology, because let's be honest, it's really, really hard. So let's go to the next slide. Yes.
Exactly. So here, so you can see actually besides SMA 200 in this screening, copolymers like ASTY 650 and also small BZ40 were also shown to be promising for cryo-EM. These 2 copolymers like SMA 200 were also identified through the step 2 thermal unfolding experiment. So here I want to introduce you to the thermal unfolding data by using the example of ASTY 650 copolymer. So firstly, you can see from nano-DSF data that we can confirm that the protein was folded and had a melting temperature around 54 degrees Celsius. And secondly, from the turbidity data, you can see that the signal is not increasing throughout the whole thermal ramp. And finally, from the cumulant radius, you can see that the increase in size did not start only at around 75 degrees Celsius. And this indicates the disassembly of the nanodiscs happened only after 75 degrees Celsius. So altogether, we see for this copolymer sample, not only the embedded protein had a great conformational stability, but also the whole nanodisk had great colloidal stability, which is exactly what we are looking for.
Yeah, that's absolutely right. We really have to remember that the most successful results comes really from this broad initial screening. I cannot emphasize this enough. And this is then followed by systematically narrowing down, like step by step, like Ping is describing here. So the more options to choose from, and in the end, the higher the success rate is, especially when your timeline is strict. If you want to go to the structure fast, it's always better to have 5 than 1 option, right? So that was our first example, already very, very insightful, but it's not everything we have to show you, because now we're going to continue with an additional case study, and that's going to further highlight how these systems are working. So what we see here is a PglB. It is from Neisseria gonorrhoeae, and unlike WbaP, PglB is a bifunctional membrane protein. So this means it has both a transmembrane glycosyl transferase domain, but it also has a large soluble acetyl transferase domain. This is what you can see up here. This is the big domain. And this dual nature actually makes it more complex, because not only structurally, but also like during the whole screening process. It makes it particularly interesting for us, because from a stabilization perspective, we're not only dealing with the membrane extraction challenges we always have to get it out of the membrane and retain this hydrophobic core, but we also have this dual domain folding dynamics. And as we're going to see here, like the presence of this large soluble domain can really significantly affect thermal and colloidal stability profiles.
That's right. So if we go to the next slide.
Yes, just a second. Here you go.
So you can see the same workflow was used also for PglB to select the optimal copolymer candidate. And here using Panta, you can see that from the step 1 size analysis, comparing the hydrodynamic radius allows the triage of copolymers that give larger particle sizes of around 20 to 30 nanometer. And then the thermal unfolding analysis gives a step 2 selection of the optimal candidate that has the highest thermal stability for both the embedded protein and the whole nanodisk. So here you might have already thought, why the onset of turbidity was chosen as a parameter to evaluate the thermal stability of the protein. So the reason is that for some membrane proteins, when the tryptophan and tyrosine residues are embedded in the high lipid content of the nanodisk, the changes in the hydrophobicity of these aromatic residues during the unfolding can be limited. And as for this situation, such as this PglB, nano-DSF signal may not be conclusive enough to understand the unfolding of the protein. And for these special cases, the multiplexing optical methods on Panta show their strengths, as, for instance, we were aware that without the embedded protein, breaking down the nanodisk along the thermal ramp alone does not lead to this increase in turbidity / formation of large amorphous aggregates. We think that the unfolding triggered aggregation of the embedded protein's soluble domain is what made the turbidity went up. So when we see an early increase in the turbidity signal, we can deduce that the protein in this sample has less optimal conformational stability, and therefore, by looking into both onset temperature of turbidity and cumulant radius, we can still select the top copolymer candidates based on the thermal stability of both the protein and the whole nanodisk. And as an example here, you can see how ASTY 1145 was more thermal stable than ASTY 650, considering both aggregation of the protein and sizing increase of the whole sample.
Yeah, that's really, really interesting, especially that the difference between those two copolymers is only their size. ST 650 is a little bit smaller, so 6 kilodalton in the monomeric version, and the 1145 is 11 kilodalton. So that's really, really, really interesting. It's also worth highlighting here that when we would look at the SDS page, we don't have it on the slide here, but those showed really like clean solubilization, clean purification across a broad number of copolymers. And the Panta really, really helped us to narrow down those massive findings to just a few, including the highlighted ASTI 1145. And those showed not just thermal resilience, but also really excellent monodispersity. And in DLS, this is something I always find fascinating. We get super monodispersed particles, something I wouldn't expect with a soluble protein, actually. So this is really, really cool. And those hydrodynamic diameters were here also in the 10 to 12 nanometer range, and our PDIs was below 0.3. So that's exactly what we want when we prepare a sample of this size for freezing on a cryo-EM grid.
Exactly. So this kind of data gives you actually the confidence to proceed. And without this early screening insights, you might have picked another copolymer based on solubilization alone and only see later in cryo-EM aggregation or broad particle distribution.
Yeah, this is exactly what we had many years. Yeah, so really, just to emphasize this again, it's not about finding a copolymer which merely works to stabilize your membrane protein. You really want to find the one that offers structural compatibility, thermal stability, and low heterogeneity. So you really have this XYZ delivered across all the metrics here. This is what we're looking for.
Yeah, definitely. And in the context of such a challenging target as PglB, the multiplexing optical methods actually allow you to always have a data-driven approach to select the copolymers.
Oh, yeah, absolutely. This makes my life in the lab easier. I have data to rely on. Okay, great. So this was already a lot of data, I know. It's a lot of biophysics, but we have one more example for you, and I promise you it gets easier over time. I went through the same. So now let's talk about our last target. It's PglC. It's the smallest and also perhaps the trickiest of the three proteins that we're covering today. Because this enzyme from Helicobacter pylori is only about 20 kilodalton in size, and it contains a single re-entrant helix. So it's not a real membrane protein which goes through the membrane, but a membrane protein which basically attaches to the membrane and partly sinks in there. And this makes it one of the most compact membrane proteins that you can work with. On paper, I know you would say, well, that's easy, it's small, but in reality, this small size is exactly what it makes so challenging for the structural studies. Proteins like this tend to have low signal and cryo-EM, and slight aggregation or polydispersity can completely derail all your efforts.
Exactly. So to my knowledge, for single particle cryo-EM, the size threshold is generally around 50 to 60 kilodalton, and anything smaller becomes extremely difficult to align and classify due to the insufficient contrast. And this means for protein like PglC, even with a perfect nanodisk, we will probably still have to add a mass increasing binder like a nanobody, a Fab, or some scaffold protein to make it visible in the cryo-EM and maybe also traceable during processing, right?
Yeah, absolutely right. So this is the one thing is that you need something to train your algorithm on, something to attach it to align your particle, and also you need a certain size. You're absolutely right. 60 kilodalton is right now the threshold, and then a Fab or a nanobody or something which increases the size over the threshold is the way to go. So you would first do this little screening process, then you would check which is the right fit, you upscale, you purify further, polish, you can do a SAC, and then bring the sample to structural quality and quantity.
Exactly. And this approach really saves a lot of time because instead of jumping into this large scale preps with 32 different copolymers, you can actually narrow it down early on with 10 microliter of screening volumes. And then you can scale up the most promising candidates and optimize further.
Yes, absolutely. That's definitely a great advantage. Okay, let's jump back to the data. So if we look at the SDS page here, we see, oh, that's beautiful. Huh? There's so many positive results. We can see that several copolymers here were able to extract this PglC, including SMA200, again, the SMART BZ35 and ASTI 650. But again, so it was the Panta that allowed us to distinguish the good from the great here again.
Exactly. And using the same workflow as we mentioned before, the SMA 200 stood out immediately from the data table. You can see that the DLS data reviewed the right particle size, also with an excellent PDI value of this peak. Actually, we did not show here, but it is below 0.25.
Yeah.
So this indicates a very good monodispersity, and that's exactly what we want to see for a protein like this.
Yeah. Additionally, we can add that the nano-DSF fluorescence ratio also showed a clear and clean unfolding transition at around like 59 degrees. So that's impressive for a protein of this size, especially if we're talking about that this is completely detergent-free and directly extracted from the membrane.
Yes. And we also looked at, sorry, could you?
Oh, sorry, Beck, I was
Yeah, so actually I just want to also emphasize that you also can look at the cumulative radius change along the thermal ramp. And this copolymer sample showed almost no signal increase before 75 degrees Celsius. This is an indication for really great colloidal stability. And again, maybe to just sum up the workflow, we use RH, so hydrodynamic radius value from the size analysis as the first selection criterion. And then we move to the step 2 thermal unfolding analysis, which have let us figure out the conditions giving both good conformational stability of the protein and colloidal stability of the nanodisk.
Okay, I have a question right about now. So can I always do it like this, or are there different options? So at first, it was really, really challenging for me to understand all those parameters since I'm not a biophysicist myself. I'm a biologist, cell biologist by training. So what's your take on this?
Yes, that's a very, very good question. And I mean, what we show here is only one way of looking into the Panta data. It is definitely up to you, the scientists, to look at the different profiling and find out how to best select among the parameters to screen for the right conditions, in this case, copolymers for your particular membrane protein. And maybe let's also use this example of PglC. We can also maybe highlight how the subtle differences can be actually detected by combining different stability parameters. So
yes. Now it's time.
Exactly, we have this nice slide showing you the subtle differences. So in this slide, we just selected three very similar copolymers, namely this SMALP BZ35, ASTY 645, and the 55. Super long names, that's why I index them for now. So
it's always hard.
Exactly. So
they were all good choices in SDS page, just to keep this straight. So in step 1 analysis was really, really good.
Yeah, exactly. And when it comes to the size analysis, again, you see that they all show good hydrodynamic radius values. But actually, it's a thermal unfolding profiling that really tell the less obvious but important differences in the stability attributes. So let me take you to a walk through this data. So actually, when you compare the candidates with very similar unfolding profiles, such as the index 3 and 2 shown in blue and yellow, you can see actually the turbidity of copolymer 3 shown in blue actually performs better than copolymer 2 shown in yellow, with no aggregation throughout the thermal ramp. And if you have got like two similar sizing or turbidity profiles, like index 1 and 3 shown in pink and blue, you can see that from the unfolding profiles that the copolymer 1 in pink has a higher melting temperature than copolymer 3 in blue.
Yeah, this is good that you use the colors. That's super interesting. So you could say that as we see from those examples, what looks good in one way might look very different in another. Can you say it like this?
Absolutely. So one only by seeing these overall behaviors, one can actually rank that copolymer 1 is best among 3 for this protein.
Yeah, that's super interesting. It's also interesting that the ASTI 645 gave promising results, like with these comparable size distributions, but it has an earlier unfolding transition. So that this tells us that they are viable, but probably not like the first choice if your goal is really going into cryo-EM, right?
Yes. So this is where these nuances matter. So for a larger protein, we might, as accept like slightly more size variation. But for PglC, it is a small protein. We really need to be extremely selective because everything from background noise to alignment depends on those nanodisk samples being perfectly defined.
Yeah, that's great. This really shows where the Panta helps us to eliminate this guesswork we scientists always have when we stand in the lab. We're so unsure what's the right thing to give to the cryo people, and then they're bothering that it's not perfect. So this really helps. So that you're just not hoping that SDS bands will translate to stable structure, but you can like actively rank and back your opinion with data. That's great.
Exactly. And what I really like is that you can make all of this assessment with just 10 microliter of purified sample. So for a low expressing protein like PglC, it is really a lifesaver.
Yeah, absolutely, that's cool. So to sum all this up, you can say SMA200 again, it proved to be a good fit for this target. But the journey to that conclusion, it always requires a complete picture. And as we've seen in many other cases, I can show you a few examples here from our own lab here. You can see that it really strongly depends on the target. So maybe for bacterial glycoproteins, SMA200 is amazing. Like for many eukaryotic targets, it's not perfect at all. So we can really just emphasize you to do the screening. It's not a marketing stunt. We really do it in the lab, and it really shows that screening is key, as I always say. So yeah, as you pointed out, Ping, for cryo-EM, we still need a binder or size increasing tech to reach this effective size threshold, but else we're really good set.
Yes. And this is why early screening followed by the scaling up and polishing is the ideal strategy. It lets you move forward with confidence, knowing your sample is both stable and suitable for the next step.
Yes. So you can wrap up the highlights and a few takeaways from today. So you can say that copolymer selection is not a guesswork process anymore. That's amazing. Together with NativeMP and Prometheus Panta, we now have really tools in our hands to make these data-driven decisions. I'm pretty sure every scientist really likes this. At least I am a huge fan. We can do biophysical profiling really early on. We can use the power of DLS, nano-DSF. We can check for turbidity, and this really dramatically reduces wasted time or time in general and efforts like on these unsuitable conditions. So for targets like PglC, it's important if your target is too small for cryo-EM, remember that you plan to add a binder, a nanobody, or anything which increases the size and helps you to align the particles later on in the evaluation process. And then best you scale up with this polishing step, which is then usually needed to reach the required quality and quantity. SMA200 proved to be highly stabilizing across different protein types for those bacterial proteins. But as I said, please don't fall into the trap and think I'm only using SMA200 now because it worked for these two proteins. I can guarantee you from more than 300 targets we screened in-house, it's not always the case. Screening is key. It's really important. Yes.
Yes. And from my side, remember Prometheus Panta gives you all this result, make it ideal for early screening when materials are precious. It is fast, easy to use, reliable, and extremely informative, and especially when dealing with low yield or structurally delicate samples.
Absolutely. Yes. Last thing, I'd like really to thank everyone who joined today. Of course, a big thank you to Greg Dodge for providing this data set. And really, this formed the core of our discussions. It's always great to collaborate with you. I don't know if you're like in the presentation or in the chat yourself. Send a hi if you are. And like he will be also part of our last part of the webinar series in part 4. He's going to make his attendance so you can see him in live also then. Make sure to register to not miss anything. And it's not over yet. We're only starting with the questions now. So I'm really happy that now we can pick questions. Kamala's going to do that for us, and then we can answer them. So, Ping, if it's okay, I just read it out, and then we can see who answers this.
Yes.
From the chat, yeah? Okay. So, great data on bacterial proteins. What is your experience with human ion channels or GPCRs, and what expression systems do you use for mammalian proteins? Okay, maybe I answer this. We have also extensive knowledge about ion channels and GPCRs. GPCRs can be really hard on expression systems. So when we have targets which are originally in human or in insects, we also try to overexpress them in human or insect cells. For GPCRs, it sometimes can be beneficial to also try this in insect cell lines. This can boost expression, but they are hard to extract. And also there, it's really important to play around with copolymer concentration, with temperature, with time, and like focus on this, and then you're going to be successful. We have purified GLP-1R, a lot of different GPCRs. We made a structure with P2X4. This is a human ion channel. It was really, really tricky in the beginning for us, but yeah, over time, I'm pretty sure that everybody can manage this with those copolymers. Okay, next question. Is there a rule of thumb for which copolymer to use? You want to give it a take, Ping, or should I? Okay, I don't hear anything from you, so I'm going to answer this also. Yeah, there's no rule of thumb. I also was hoping to find one, but yeah, more than 300 targets screened. I haven't found one so far. So I know it's frustrating, but it is really reality. Like some backbones sometimes perform better on specific target classes, but there's no real thing to pinpoint it. So this is unfortunately really hard. But with Panta, it's easier to screen. Okay, next question, please. Do you have to purify them individually using 16 different copolymers? Isn't that time consuming? What stage is this? Yes, as of today, you have to do this individually, but if you have a robot to do it manually, you can also do it manually. And we are in the process of presenting something to the community, so we are launching our own automation platform. So in future, there's much more to come. But we do it here in the lab, also by hand right now. And yeah, it's time consuming, but you can manage 16 different easily in a day. So that's possible, at least what we do here. Okay, in comparison to detergents, copolymers tend to extract and solubilize much less membrane protein. What would you suggest us to increase the yield enough for cryo study? Okay. Sorry, Ping, if you want to answer something, please feel free. I don't want to
go ahead.
Talk over you. Just on copolymer questions. For some targets, I agree with you. For other targets, I have to disagree with you. We have had targets where with copolymers, we could significantly get more protein out of the membrane than with detergents. We had examples where it was impossible for us with any detergent we tried to solubilize specific membrane proteins. And with detergent, it was a breeze. And it worked just in a day to get proper amounts out. So this really highly depends on the target. And also here, experiment with the extraction time, experiment with extraction temperature, and with the copolymer ratio. These are the three parameters I can really advise you to test, and this is going to make it successful. At least it does in our hands, in our lab. Yes. Okay. Do Panta DLS measurement also give zeta potential? That's one for you.
No, the simple answer is no.
Okay, then we have to go to Melbourne for this. Okay. Could you comment on the compatibility of these copolymers with various affinity resins, so nickel, NTA, FLAC, and streptoctin? Yes, I can do this. So in literature and from the community, we know that especially for his-based purifications, the copolymers in excess can interfere with the binding to the resin. We have developed a solution for this. It's called PglHunter. You can just buy it from our website. It's a resin which specifically catches free copolymer in solution, which you need in the solubilization step to solubilize the maximum of your membrane protein out of there. And then in the later steps, it interferes. So you just run it through the agarose, like a drip column, and then you're good to go. Another alternative is to dilute your sample at least 1 to 10, better 1 to 100. I know this is a bit unpractical for larger volumes. This is why we have PglHunter. And then this usually works way, way better. For FLAC and streptoctin, it is discussed in some targets with some people, but I have not read anything publication-wise so far now. But dilution can help. Yes, I would answer it like this. Okay, next question. Have you measured the nanodisc alone without membrane protein with the DLS from Prometheus Panta? You want to answer, Ping, or should I?
I think from our discussion, I know that you have done that.
Yeah.
And actually, there's no sizing increase or turbidity increase over the thermal ramp.
Exactly. Yeah. Over the thermal ramp, there's nothing. The nanodiscs are uniform in size. If you just fill them with lipids and form them, you are usually at around 3 nanometer in hydrodynamic radius, and they're super stable. It's nothing happening with them. Yes. Okay. Next question. Are these examples for purified membrane proteins in pure lipids? Native membranes will contain tons of other proteins. Yes, you're right, but these examples are from purified native membranes. So it's overexpressed with an affinity tag. In this case, it was a double strep tag, as far as I remember, and purified with streptactine resin from eBay Life Science. And it was directly from this bacterial membrane. So just by affinity purification, Greg was able to get these beautiful and really, really clean results. So that's definitely possible. And that's also what we do in lab here. Here in-house, we really love the Row 1D4 tag. It's my favorite tag ever in the world. Like, I go out there and tell this is the best tag, in my opinion, for membrane proteins. Yeah, and we do it exactly like this. And you can get very, very clear and very clean results with that from any kind of membrane. Next question, please. 20 kilodalton, and you solve it with cryo-EM. How? Yeah.
I think this is for the PglC, right?
Yeah, this is for the PglC. Yeah. Of course, you don't do it just like that. You add something, a nanobody, something to increase size, something to get a better view on it, to get it hooked in, and then you can also solve this. But I honestly don't know if it's solved right now. Do you know, Ping, if Greg already solved this one?
I'm not following that, unfortunately.
That's okay. Okay, next question. If my protein is not overexpressed, how can you confirm the extracted one is what I want without purification like nickel color? It's going to be very hard. What you could do is, if you have a specific antibody which can bind your protein, you could add it to an agarose or magnetic particle and pull it like there, then you would get a clean result. But else you have to do all the other options, how you can purify non-overexpressed proteins and distinguish them from the other ones. But it's going to be way, way harder. That's absolutely clear. So overexpression, if feasible, is of course easier, and it's going to give you more out in the end. Okay, we have more questions. Oh, we have so many questions, it's crazy. From Biotech. PglC further optimization. Could you also measure PglC linked to nanobody FAB with the Prometheus Panta? It's a good question.
Did you do that? Yes, I think in theory you could. I'm not sure if you would have done that in your lab.
No, we have not done this.
But
you could, and I think you could also see when it falls or if it falls off, right? You would be able to see that. Yeah.
Exactly, because actually, I mean, for the DLS, it's universal, and anyway, you would still want to have like a monodispersed sample after all. And for the nano DSF, Panta can resolve multi-domain unfolding events, so it's not a problem. Is this capillary suited for all kinds of samples, or is this the same as MST, where you have the premium capillary and normal capillary? So there are two types of capillary. The standard ones can measure sample concentration above 200 microgram per milliliter. And if your sample concentration goes below that, you would have to use a high sensitivity capillary, which is made of quartz, to reduce the background fluorescence.
So if you have very, very low expression level, then of course it would be beneficial to bump that expression level up, try something else. Our cell culture does this all the time. We also have these issues that sometimes we have barely any expression. And if you are not able to bump this up and you really, really want to purify it, you have to go up in volume and then do the volume work. But you don't have to see this in SDS page to analyze this. So Ping already explained that we have the premium capillaries you can use, and there you can also analyze your sample in less concentration, but you need a specific concentration threshold to really trust the results. So it would be great if you would have something to also work further with.
Yeah, definitely. I think if you cannot see a band in SDS page, the concentration might be really low. I cannot comment on how much you can get after the purification. So I would say maybe perhaps it's too low.
Yeah, what we were doing in the beginning was then just like take several liters and just concentrate your elutions until you reach a certain threshold to then at least analyze if it's if it's good to work on with this. Yeah, but that's an issue with membrane proteins, that's for sure. Okay. I've thought about methyl anthraquins, so additives which coordinate, which serve as tryptophan and enhance emission with an unfolding profile.
Would you like to first comment from your experience?
Yeah, I don't have any experience, so this is a very NLR-based questions, but we're also going to answer this afterwards. That's why I email. Sorry that I can't answer this. I'm right from the bat.
Yeah, I think you might have some influence in the single wavelengths, but also as nano DSF is detecting the ratio of the fluorescence, maybe from the ratio view you have a good chance to see it. Okay,
well, that's good info. Thank you. Okay, will PglHunter capture copolymer reconstituted membrane proteins, not just free copolymers? For example, will the yield of the membrane protein be reduced by using PglHunter? So we have verified that it only catches free copolymers, but you're going to have a retention effect, like a bit like a size exclusion effect inside. So this is why we described in the protocol how you have to elute it to make sure that you retain all your preformed nanodisk, that this doesn't happen. That's it? Or it's going on, it's going on. Okay, well, that's really cool. I've noticed in the first example that you used the single wavelengths for the TM value, and then in the last example, you used the ratio for the TM. Why is that? Okay, why is it?
Yes, that's actually a very good question. So in the single wavelength detection, we look at the change in the fluorescence intensity at 330 or 350 nanometer over temperature. And it is possible that the unfolding transitions which do not trigger an emission, fluorescence emission peak shift are visible in the single wavelength data, but not in the ratio. So this can be, for instance, when tryptophan residues are already surface exposed or they're just tyrosine residues, and this could be the case. So the ratio method helps us to remove actually the problem that the fluorescence is decaying with the increasing temperature, and therefore we prefer to use the ratio first. However, if the unfolding is already highly visible in the single wavelength view or only visible in the single wavelength view, it is absolutely valid to use them for the TM.
Perfect. Okay, let's do one more question and then the rest we answer via email. What's the relevance of second vial coefficient in Panta, B22?
Yes, so maybe it's not super relevant to the membrane protein research, but this is a parameter used for formulation, where you can basically work with low concentration of the protein and predict if the protein tends to interact with themselves, or let's say getting solved by the buffer at higher concentration. So you can say like you are working with, let's say, 0.5 to 10 milligram per milliliter, but you want to know how the protein is at 100 milligram per milliliter.
Great, thank you very much. Then we're going to answer the rest of all of the questions by email, and of course the questions we were unfortunately not able to answer right from the bat. And I want to thank everybody again for attending. This is already a little advertisement for our next webinar. It's not together with NanoTemper, but with Cube we have these master classes. It's going to be on the 22nd of May, so feel free to register there. And this is another super interesting topic. It's affinity selection, mass spectrometry, ASMS. And this is the high throughput screening of the GLP-1 receptor. We produced this in-house and we collaborated with Momentum Biotechnologies here. My colleague Marina is going to lead through the webinar, and the expert Sham is going to present all of this exciting data. So really, really please register if you're interested in this. I found this collaboration super cool. It was really, really interesting. And apart from this, thank you very much, Ping, for doing this together with me. It was a pleasure, as always. And thanks everybody for attending. Hopefully see all of you in the next webinar.
Thank you all for the great questions.
Yeah, it was really cool. Thank you. Okay.